Okay, let's jump straight in. Today, we're doing a deep dive into something pretty fundamental happening inside our cells. We're looking at the nuclear shuttling of H2A, H2B histone chaperones, HCs for short. Our mission really is to understand how these specialized proteins, these HCs, manage to get histones, which are, key structural proteins for DNA, how they get them from the cytoplasm, where they're made, all the way into the nucleus. That's where the action is, where chromatin gets assembled.
Right, it's like a microscopic high-stakes delivery service. These HCs, you can think of them as molecular bodyguards for the histones. They stick with the histone, preventing it from clumping up or sort of interacting incorrectly as it travels. But the real challenge is crossing that border, the nuclear envelope, through something called the nuclear pore complex, the NPC. And that move isn't random. It requires active transport, coordinated, using very specific signals. Address labels, basically.
Address labels. Okay, that makes sense. So let's unpack that. What are these signals, these cellular traffic signs that the HCs use?
Well, there are primarily two types recognized by carrier proteins. You've got nuclear localization signals, NLS, which says, okay, go into the nucleus. And then nuclear export signals, NES, for the opposite journey, getting out. In this study we're looking at, it specifically tried to predict where these signals were located on 9 important H2H, H2B chaperones. Proteins like NPM3, some heat shock proteins.
Ah, okay. And here's where I remember it getting really interesting. The predictions about which signals they use. NLS signals, they come in two types, right? Monopartite and bipartite.
That's right. And for the import signals, the NLS, the prediction software, showed most of these chaperones prefer the bipartite kind. Think of it like needing 2 parts to the address label, separated by a little space. It's generally a stronger, more specific signal for getting into the nucleus. For instance, one called VPS72. It showed really high predicted NLS activity, scores of 10 and 11, which is basically off the charts. Suggests it's meant to stay inside the nucleus most of the time.
Wow, okay, so VPS72, maybe like a core resident, is strongly signaled to stay put. But what about the others? Did the export signals, the NES, tell a different story?
Oh, absolutely. A very different story for some. The heat shock HCS, those are HSP90A, HSP90B, HSP7C. They were predicted to have way more NES sequences compared to others. Proteins like SB16H, for example, had 0 predicted NESs. So this suggests the heat shock proteins aren't just hanging out inside. They need to shuttle in and out dynamically, maybe like first responders dealing with cellular stress.
Right, rapid response team. Okay, so we have these predictions for specific address labels, specific roles, dynamic shuttling for some, more residential for others. But if the signals are predicted so clearly, why did the study then highlight a problem? This seems to be where the picture starts to break down.
Exactly. This is what the researchers called a key lacuna, a major gap in our knowledge. Because the software, based on known rules, predicted where these proteins should be located within the cell. But when they compared these predictions to what's actually been observed and published in experiments with living cells, there were significant conflicts. Like, the software predicted NPM3 should be mostly out in the cytoplasm, but lab work consistently finds it predominantly in the nucleus.
Huh. That's a pretty big mismatch. It's like the GPS saying you're in the ocean when you're standing in the desert. So that disconnect, it implies our understanding of how this traffic is regulated is, well, incomplete, doesn't it?
Precisely. It suggests just having an NLS or an NES sequence isn't the whole story. There must be other layers of control. And it raises a really important question. These HCs must be doing more than just, histone delivery for building chromatin. Their ability to bind histones likely connects them to other jobs, maybe linking up with other regulatory pathways in the cell. They could be acting as sort of integration points.
Okay, so bringing this all together, what's the main takeaway for you, the listener, trying to make sense of this cellular complexity?
I think the main thing is that the histone chaperone story is, well, it's complicated. We really need a broader view, combining different approaches to understand the big difference between what we see HCs do in a test tube in vitro versus inside a living cell in vivo. There's clearly a gap there, and figuring out why the predictions don't always match reality means rethinking how these proteins are actually managed in the cell.
That really highlights the challenge, filling that knowledge gap.
And it's not just academic. Understanding these molecular details, resolving these discrepancies, it has real potential significance. Consider a protein like NPM2, it's another chaperone, and it's absolutely essential for the very first cell division after fertilization in mammals. That jump from 1 cell to 2. So if we can figure out why its a predicted location or behavior might differ from reality, if we truly understand its regulation, that could potentially open doors for understanding and maybe even treating certain types of human infertility. It's fundamental biology with potentially profound applications.